next witness?
Yes, Your Honor. The Commonwealth will call Mr. Nicolas Bradford to the stand.
Step right up. Just watch your step for me.
You solemnly swear the evidence given to the court and jury in the case now we're hearing the truth, the whole truth, and nothing but the truth, so help you God.
I do.
Thank you.
Forgive me, Your Honor. May I have just one moment?
Sure.
Good afternoon, sir.
Good afternoon.
Could you please state your name and spell your last name for the jury?
Yep, it's Nicolas Bradford, Bradford.
And sir, that microphone before you is adjustable. You can place it anywhere that's comfortable for yourself. Okay. What is it that you do for work, sir?
I'm a DNA analyst for Bodie Technology in Lorton, Virginia.
Okay, you're definitely going to have to keep your voice up, sir, okay?
Sorry about that.
You,
Now, sir, if I could.
Sorry. It's Bodie Technology in Lorton, Virginia.
And for the record, Bodie is just spelled as far as B-O-D-E. Is that correct?
Correct.
Now, sir, if I could just ask you some questions about your educational background, starting with your undergraduate. Where did you go for your undergraduate, and what, if any, degree did you receive from there?
Yep. I have a Bachelor of Science in Forensic Biology from Mercyhurst College in Erie, Pennsylvania.
And then, following your graduation with that degree, where did you go?
I have a Master of Forensic Science in Forensic Molecular Biology from the George Washington University in Washington, D.C.
And whenabouts was it that you received that master's?
It was around about 2011.
And with relation to your graduate work and your work at Bodie, when did your work at Bodie begin with relation to your graduate work?
I started at Bodie at about 2009.
So during the course of your graduate school?
Yes.
And when you started with the Bodie lab, what was the sort of position or what did you start doing when you began?
I was a DNA technician. We assisted the analysts with their case files, and we primarily operated the different robotics and the different processes in the lab.
And at some point over the course of your work there, you then became an analyst, is that correct?
Yes.
And about how much time elapsed, or how much time did you spend as a, excuse me, as a technician prior to becoming an analyst?
I was a technician for about a year, maybe two. And then I was in the data banking section as an analyst. And since then, I've worked in the high throughput casework group and the customized casework group.
Now, as far as your work within this particular field, have you had occasion to work at any other sort of labs or facilities?
Yes, for about 3 years, I worked at the Pennsylvania State Police DNA lab.
Now, Bodie Technology, as far as that lab is concerned, can you describe for the jury sort of what kind of lab that is? And let me ask you first by just about how many analysts work at Bodie Technology?
Somewhere in the neighborhood of maybe 100 analysts, give or take. We're a small private lab in Lorton, Virginia, and we do DNA work contracted through different police agencies throughout the country.
Now, in addition to police agencies, are there other types of entities that you would accept or that would send submissions for DNA casework?
Yes, we do some international work with human identification and that sort of thing as well.
Now, as far as the lab at Bodie Technologies, is that lab accredited?
Yes, we are accredited through the National Standards Institute.
And can you speak a little bit about that sort of accreditation process and what it means to be an accredited lab?
Yes, so that the National Standards Institute has a set of criteria for DNA labs doing forensic work, and we are audited against those criteria every year, sometimes internal audits and sometimes external audits.
Is that accreditation at the lab, is that up to, the accreditation, is it current as of now?
Yes.
Now, with reference to your work as a DNA analyst, can you describe, just in general terms for the jury, what it is that you do in reference, what is it that you do in reference to your work as a DNA analyst?
Yes, so we take in evidence that's submitted to us. We process it through the DNA processing. So there are 4 basic steps to DNA. There's extraction, so that's where we separate out the DNA. And then there's quantification, and that is basically just us determining how much DNA we have to work with. The next step is amplification. So depending on the case and the different processes that we're going to use, we're targeting a number of different locations in the genome, and we're going to make millions of copies of each of those locations. And in each one of those copies, there's a little fluorescent tag that we can then detect in the detection step, and that's the 4th step. And then that, from there, we have the DNA profile for that sample.
Now, with reference to the lab, as far as training is concerned, you mentioned that you spent at least a year or 2 as a technician prior to becoming an analyst. What are some of the training, specialized training in this area that you wanted to go as an employee at this lab?
Yes, so the training program involves a lot of scientific literature reading. There's a practical aspect to it where we're given known samples that we're to treat as if they were casework samples. So we process those as if they were casework. We have written and verbal exams, and we have a moot court exam at the end.
And you successfully completed each of those training sort of portions of, over your course of work with the lab?
Yes.
Are you familiar with a term known as a proficiency test?
Yes.
And can you explain to the jury what that is and how it relates to your work as a DNA analyst at Bodie Technology?
Yes, so part of our accreditation is we have to do proficiency testing twice a year. And that is when an outside company sends us known samples and we process them, again, as if they were casework. And then those results are sent to that outside company, and then we get the results back to see if we are proficient in those technologies or not.
And with respect to yourself, as far as the proficiency testing that you've undergone, what, if any, issues have you had with proficiency testing that you've undertaken?
I've passed all my proficiency tests.
Now, what is DNA?
So DNA is essentially the blueprint for a living thing. It's in most of our cells, and it's unique to an individual with the exception of identical twins.
Now, at some point in January of 2024, did you receive a particular assignment in regard to DNA analysis from a number of items that you received from Massachusetts?
Yes.
And specifically, can you describe the process as far as like when an item comes into the lab at Bodie Technology, how it's sort of processed, or how from the time of receipt to the time that the item gets to you? Can you explain a little bit about that process?
Yes, so the item is generally sent via UPS or FedEx. It arrives in our secure evidence room. That room has limited access to only certain individuals. And from that point on, when it's received, a chain of custody is started for that shipment. And anytime anyone touches or uses that item, we need to check it out from the secure evidence room. And then that's all logged on the chain of custody.
Now, when something comes in, is it assigned a sample name or a case number or an ID number in some way, shape, or form?
Yes.
And is it also identified by sort of the corresponding numbers from the external vendor where it came?
Yes.
Now, in reference to Bodie case number CCA 2416-0023, did you perform work in relation to that case?
Yes.
And there was a corresponding agency case number of 22-02184, is that correct?
Yes.
And that would have been from the outside vendor, or at least one of them, as far as the Massachusetts State Police crime lab, correct?
Correct.
So there were a number of items that you received. If you could identify them for the jury, what you received and specifically sort of what you looked at.
Yes, so we received an apparent hair. We received an extract from a taillight from a vehicle, and we received 2 reference samples from 2 troopers from the Massachusetts State Police.
And in addition, was there a sample ID as far as a known standard?
Yes. So Bodie gives it our own our own item number. So in this case, the 2 reference samples were CCA 24160023 R03 and the same number R04 for the 2 reference samples.
Now, if I could just ask you to further expound upon as far as the CCA 2416-0023, which is E01. There was an agency sample ID, is that correct, that was attached to that?
Yes.
And what was that, sir?
So that was 3-6.1.
Is there a description that comes from the external vendor as well as to sort of what that item is?
Yes. In this case, the sample was a shaft end of hair from exterior passenger side rear panel.
There's a specific mass registration plate that's associated with that hair as well, is that correct?
Yes.
And that was 3GC684, is that correct?
Correct.
Now, with reference to the R03 and R04, those were vehicle samples, is that correct?
Yes.
And R03 specifically, as far as the agency description, what, if anything, was provided as far as that?
For R03, that was the buccal swab from Yuri Begetic.
And as far as R04?
That was the buccal swab from Michael Proctor.
Now, with reference to the extract portion, that was E02, is that correct?
Yes.
And if you know when you say extract, can you explain to the jury sort of what you understand that term to mean, or what is an extract as far as what you looked at?
Yes. So when I described the 4-step process earlier, so the extraction was already done prior to us receiving the item. So essentially, the extract comes to us in a tube, and it's a liquid. And provided with that would be the negative control that was associated with it. So from that point, we have the quantification information from the lab that did the extraction. So then we would amplify and then detect the profile from there.
And just to be clear, as far as the E02 that you were talking about, as far as the extract that you had received, what was the agency description on that extract?
So that was the sample from passenger side taillight with the same registration number, and that was labeled as an extract.
Now, in addition to that, there was a known blood standard, which you indicated was item or agency sample ID 8-1.1.1.
Yes.
And what was the agency description on the known blood standard?
That would be from, excuse me, sorry, that was the sample from John O'Keefe, the profile, I should say.
And so when you say the profile as what do you mean by that?
Yes, so the profile was generated by the Massachusetts State Police Lab, and then that profile was given to us to do a comparison.
Now, as far as the profile that was provided, as far as your observations of that, what, if any, issues did you observe in your observations of the profile that was provided by the State Police Crime Lab in Massachusetts?
It looked like a standard single source profile.
Now, starting with, as far as there was a STR processing that was conducted, correct?
Yes.
What does STR stand for?
It stands for short tandem repeats, and essentially what we're doing with that is it's the DNA process that we use in forensic testing, where the areas that we're amplifying are short repeats of the genetic sequence, that repeat multiple times, and we're just measuring the number of times that sequence repeats. And those numbers together are the profile.
Now, as far as the profile from the, excuse me, the profile from the extract that you received as far as EO2, excuse me, from the passenger side taillight of the vehicle, what, if anything, were you able to determine based on your analysis and SDR processing of that?
Yes, so we determined that it was consistent with a mixture of 3 individuals, including at least 1 male contributor.
And so, if you could just walk the jury through as far as the process, as far as the testing, what is it that you're actually doing in the lab? What are you looking at, and how are you making a comparison?
So once we have the DNA profile, like I said, we're measuring the number of times the sequence repeats at a given location. So we have a series of those, and then we compare that to the reference profile that we were provided, or that we generated for the 2 reference samples that we actually processed. And then we determine if it's possible that the individual whose reference profile it is could be a possible contributor to that mixture profile that we got from the evidence. And then we need to do a statistical calculation to determine the strength of that possibility of an inclusion.
Let me ask you a little bit about, as far as that statistical calculation, how is that done, or where does that come from?
So in this case, we did what's called a likelihood ratio. Essentially, what we're doing is comparing 2 explanations for the DNA profile that we observed. So if we have, say, person A, one explanation would be that the DNA profile is a mixture of person A and 2 unknown individuals. And then the other explanation would be that it's simply 3 unknown individuals and it's not person A. So then we compare those 2 explanations of the profile and determine which one is more likely.
Now, as far as the sort of reference samples that you had the DNA profile of John O'Keefe, correct?
Correct.
And I'm sorry, before starting that, you had the extract with the DNA profile with multiple contributors from the tail leg, correct?
Yes.
So then you have the known blood standards or the DNA profile from John O'Keefe, correct?
Yes.
And then you have the buccal samples from Yuri Buchanan and Michael Proctor, correct?
Yes.
And so with reference to those specifically, as far as the reference samples, is that what you're generating the DNA profiles from?
So we're comparing those references to the mixture that we obtained from the taillight. Yeah.
And so in your comparison, let me start with the known one as far as John O'Keefe. What, if anything, did you find from your comparison of the John O'Keefe DNA profile to the DNA profile from the taillight?
Yes. So in this case, when comparing the John O'Keefe profile, assuming a mixture of 3 individuals, it is at least 740 nonillion times more likely to be observed if it originated from John O'Keefe and 2 unknown unrelated individuals than if from 3 unknown unrelated individuals. And we provide a sort of verbal qualifier to that because most people probably don't have a lot of experience with nonillion. So in this case, that would be very strong support for inclusion.
If you know nonillion, how many zeros are we talking?
So that would be the number with 30 zeros.
And so that would be a strong support for inclusion, correct?
Yes.
Now, with regard to your comparison, with regard to your work as a DNA analyst, is there any kind of software that you use in the course of your analysis with regard to an SDR mix?
Yes, when there's a mixture profile, we often use what's called StarMix, and it's essentially a computer program that we input the DNA profile, and it allows us to use a series of mathematical equations to help us interpret that mixture.
And was that done in this case as well?
Yes, and then we use that same program to run the statistics.
Now, with regard to the sample, the buccal sample attributed from Yuri Buchanan, Did you run that for a comparison as well?
Yes.
And what, if anything, were your findings in relation to your analysis of that?
So in that case, assuming a mixture of 3 individuals, the DNA profile obtained is at least 46 million times more likely to be observed if it originated from 3 unknown, unrelated individuals than if from Yuri Begenik and 2 unknown, unrelated individuals. So that sounds confusing given what I just said. So the verbal qualifier for that would be very strong support for exclusion.
I'm sorry, very strong support for exclusion, is that correct?
Correct.
And then similarly, as you were testifying, what, if anything, did you find from your analysis comparison of the buccal sample or DNA profile for Michael Proctor?
Yes, so in that case, assuming a mixture of 3 individuals, it is at least 76,000 times more likely to be observed from 3 unknown, unrelated individuals than if from Michael Proctor and 2 unknown, unrelated individuals. So that would be strong support for exclusion.
So correct me if I'm wrong, but your findings with regard to the 3 samples is strong inclusion or strong support for inclusion of John O'Keefe, is that correct?
It would be very strong support for inclusion.
And strong support for exclusion of both Yuri Begenik and Michael Proctor, correct?
Correct.
Now, sir, in addition to that, you had mentioned that there was an E01 sample that was a shaft end of a hair from the exterior passenger side rear panel of that same vehicle with the same Massachusetts license plate, correct?
Correct.
Now, with regard to DNA as it pertains to hair samples, can you describe for the jury sort of that process and so what is it that you're when you receive a hair sample as far as an extraction is concerned, how does that process work or what are you getting that extraction from?
Yes, so when we have a hair, generally we'll take a look at it and a forensic biologist will give their opinion on if it's likely that we'll obtain a profile from that hair because hairs can be a little tricky. So in this case, the forensic biologist determined that the hair would likely not provide a great profile from STR testing, but we went ahead with it anyway, just because sometimes it does work. But in this case, we extract it, and that involves dissolving the hair and forming an extract like the other samples. And then we quantified the hair in this case, and the quantification that I used targets what is called autosomal DNA or nuclear DNA. And in that case, the results were below the limit of detection. So we did not process that sample forward for STRs because we would not have obtained a useful DNA profile from it.
Now, sir, if I could just take you back a little bit in that. Earlier in your statement right there, you just indicated that hair samples or DNA from hair can be a little tricky. Can you expound upon that and explain to the jury sort of what you mean by that?
Yes. A lot of times, hairs are, unless they have a root attached, they're not a great sample for nuclear STR testing. They can be, but it's sometimes hit or miss.
As far as what's actually being tested as far as STR when it comes to hair, it's not the actual hair, is that correct?
I'm not sure I follow. Can you?
Let me ask you this. You're familiar with the term follicular tissue?
Yes.
Okay. Can you explain to the jury what that means in relation to a hair sample and DNA testing?
Yes. So when there's a follicle attached, that's generally where you will get the nuclear DNA from, not the hair itself.
And the follicle would necessarily be attached to the root end and not to the shaft, correct?
Correct.
Now, as far as autosomal DNA, can you explain what is autosomal DNA and sort of where does that come from within DNA testing?
Yeah, so you can think about that more as nuclear DNA. So that would be DNA that's in the nucleus of the cell. So that's what most people think of when they think of DNA. But there are there's other forms of DNA. So like, for example, mitochondrial DNA that exists outside the nucleus.
And as far as the testing that you performed in regard to this hair sample, that was STR testing, is that correct?
Yes.
And I'm sorry, what were the findings again with relation to the SDR test?
Yes, so like I said, we quantified the hair, and it was below the limit of detection, so we did not process that hair forward for STR.
Now, as far as that terminology, below the limit of detection, can you explain a little bit to the jury sort of what that means as it applies to the testing that you conduct?
Yes, so the instrumentation has a limit of detection, so there could be potentially small amounts of DNA there, but it's below the limit that we are confident to say that there is DNA present. So we would normally not process those samples forward.
Now, that hair sample, as far as the one that you tested, you mentioned that it was not initially advisable, I guess, for STR testing, and you just went ahead and did it anyway, because sometimes it works. Is that correct?
Yes, it's usually worth a try.
And then beyond that, that hair sample within your lab, as far as Bodie technology, was that given to a different analyst for a different type of testing?
Yes, we decided it was worth a try to do mitochondrial DNA testing on the hair.
And is that a type of testing that you yourself perform?
No, that would not be me.
And why would it not be you?
I was not trained in mitochondrial testing.
That's sort of a specialized subset as far as different training and different type of testing that occurs?
Yes.
Just in general terms, if you know, could you explain to the jury sort of the difference between what you've termed as nuclear or autosomal DNA versus mitochondrial DNA?
Yes, so very basically, the nuclear DNA is the DNA that exists in the nucleus. The mitochondrial DNA is the DNA that exists outside the nucleus in the mitochondria. And the mitochondrial DNA is inherited directly from the mother. So if you were to test maternally related individuals, they would have the same mitochondrial profile.
And that other analyst that conducted the mitochondrial DNA, are you familiar with who that person is?
Yes, that was test chart.
Okay, one moment, Sean.
Sure.
Thank you very much, sir. I have no further questions.
Okay,
We have no questions.
great. So you are all set, Mr. Bradford.
Thank you.
Thank you very much.